Good morning, everyone, and thank you so much for the opportunity to present my work. So my work is titled Spatiotemporal Perturbation of the T-cell Actin Cytoskeleton, and I'll talk about how we did this using a novel tool that we developed. So I'll jump right in. Why T-cells? The T-cells are an extremely important component of the immune system and perform the very crucial task of immunosurveillance, where they scan the body for signs of danger so that they can eliminate it and protect the body. And throughout all these different steps of immunosurveillance, such as extravasation, migration, and forming the very crucial cell-cell contact interface known as the T-cell immunological synapse, there are a lot of unique cell biological demands that the T-cell faces. And the T-cell actin cytoskeleton actually matches these demands by forming a lot of specialized structures and aiding in, say, acting as a scaffolding during signaling, post-generation, mechanosensing, and many other functions during many steps of immunosurveillance. So looking at this versatility of actin, one might ask, how does the same unit network of actin perform so many diverse functions? The answer may lie in the diversity of architectures of actin as well as the diversity of their dynamics, both of which are seen at the T-cell immune synapse and at different stages of T-cell immunosurveillance. And in fact, genetic lesions in a lot of these actin-regulating proteins result in either lethalities or debilitating immunodeficiencies. So this really highlights why we need to study actin in immune cells. So let's study actin, right? The issue here is that the actin cytoskeleton is very challenging to study, particularly so in T cells, because the T cells are highly small, they're round, they have a very tortuous surface with a lot of projections, they're very fast moving. They're hard to transfect with genetic probes, they have very rapid cytoskeletal dynamics that are hard to track, and they actually have a lot of common regulators for diverse actin architectures, an example being the ARP2/3 complex, which is a very important branched actin nucleating factor. And it is responsible for the functioning of a lot of these actin foci that you can see, as well as the branched actin networks in the periphery. And so how can I tell what the function of one structure is versus the other without ablating both? Because common techniques that are available now, like genetic methods, antibodies, and so on, actually would ablate actin across the entire system at all of the time, and I would lose that resolution to look at simultaneously coexisting actin structures. So how do we move beyond this technical limitation so that we can establish a clearer structure-function relationship between different actin architectures and their functions? And how do we achieve the precise spatiotemporal resolution required to perturb actin polymerization in this way? So we thought we could use light because we can control where and when light falls upon a sample. And the azido group actually is a very well-known chemical moiety that is covalently photocross-linkable. And using it, we designed and characterized our novel light-inducible inhibitor of the ARP2/3 complex, which we call AZ. So the current state of art is CK666, which we all know about, which binds to the ARP2/3 complex in a pocket and prevents nucleation of new branches of actin. But we don't have any spatial or temporal control when we're using this. It's highly reversible and it limits the scope of its use in a lot of cell-cell conjugate systems. It has a very high IC50, so you have to use a lot of the compound, which can lead to unwanted side effects and even some off-target effects. So how do we face these challenges using AZ and use it for the studies that we want to do? So imagine this is a cell and you have ARP2/3 complexes producing new branches of actin. CK666 at a relatively higher concentration, which is we're just using as a benchmark here, can ablate all of these complexes successfully in the cell. However, if we want to establish a particular relation of a structure-function relationship here, we can use AZ because we can push the equilibrium towards the bound state using light at a particular region at a particular time in the cell, leaving the others untouched, and we need relatively lower concentration to do so. So now with AZ, we have spatial and temporal control, we have improved binding because there's covalent cross-linking involved, and there's an effectively lower IC50 at the binding site. We also have seen that there's irreversible binding within certain timescales of our studies, and nice for us, which is we have a novel inexpensive synthesis scheme, so we can produce a lot of the compound and in a pretty cost-effective manner. So now that we have AZ, the first thing we wanted to check is, does it actually work? So we used an in vitro actin polymerization assay to do this. where in a tube we took pyrenyl actin, which is actin, G-actin, labeled with quenched fluorophore, and then when these actin monomers are added onto a filament, the fluorophore dequenches and there's fluorescence. So fluorescence is a readout of polymerization. We also added purified ARP2/3 complex protein and AZ, our compound, and photoirradiated the mixture and measured fluorescence over time. And you can see in the case of AZ and light treatment, there is a significant decrease in polymerization, indicating that the compound works. Interestingly, we also saw that the compound in the absence of light has no effect whatsoever on polymerization. This would tell us that there is a light-dependent switching in its activity that can add an additional temporal control to its function. So now we know it works in vitro, but does it work inside cells? That's what we did next, using Jurkat T cells that were suspended in media containing AZ and then photostimulated, and then allowed to adhere to ligand and antibody-coated cover slips, forming immunological synapses. And then we measured the level of actin at the interface using fluorescence microscopy. And in fact, we'll see in the case of AZ and light treatment, there was the most significant decrease in actin levels, indicating that the compound works inside cells. And we also replicated this in primary T cells that have notoriously fast actin dynamics. And here too, we see a decrease in the level of actin, as well as the level of the well-known branch point marker, phospho-HS1, indicating it's working in these different types of T cells. So looking at this whole cell perturbation, we actually got very curious as to where the loss in branched actin is the most pronounced at the T cell synapse, because that could tell us about where the inhibitor is incorporated the most and where ARP2/3 complex dynamics is the highest. And so we use structured illumination microscopy of Jurkat T cell synapses to answer this question. So to reiterate, this is a Jurkat T cell immunological synapse, and it's characterized by this very typical peripheral actin ring, which can be divided into the lamellipodia, the lamellar region, and the inner actin structures. And we similarly segmented our synapses in this way. And when we observe the lamellipodial region, we see a distinct dimming of actin networks in that region upon AZ and light treatment, indicating that that may be the region of higher ARP2/3 complex dynamics, leading to a higher incorporation and effect of this inhibitor. This was also corroborated by our scan electron microscopy data of similar Jurkat T cell cytoskeletons that we extracted using a detergent-based method. And in this peripheral region, particularly towards the lamellipodia, we see a distinct decrease in branching density or network density, indicating that this is what the whole cell effect and where the incorporation looks like. So now we know that at the whole cell level, this is how the compound works. So now we moved to our original purpose, which was spatially defined perturbation. And we performed an assay at the population level where we can essentially pick out which cells we want to perturb in a milieu of cells. And so we used a migration assay where we allowed T cells to migrate around on a coated cover slip in media containing our compound, and then we photoirradiated and ablated only half of the field of view and left the others untouched. And then we followed these cells for up to 15 minutes and tracked their trajectories. And very interestingly, we see that a single short pulse of ARP2/3 complex inhibition was actually enough to slow down these primary T cells migrating around with respect to their total distance, the mean speed of their trajectories, as well as the time they spent stationary. So from the population level perturbation now, we moved to the holy grail in live cell imaging, which is subcellular perturbation. And here too, we used a T cell synapse model, where in fact it's known that if you perform localized perturbation in the periphery, it can actually destabilize the synapse architecture. And we saw something similar in our system here, where we have Jurkat T cells expressing LIFAC citrine, which marks their actin cytoskeleton. And these cells were allowed to synapse actively on a cover slip in media containing our compound. And then in live mode, we ablated in this region, as you can see. And you'll notice that there are some sort of filopodia-like pointed structures arising, which is quite typical of ARP2/3 complex inhibition, as well as a symmetry change, which you will then observe a little better in these examples as well, where you can note the symmetry change and the appearance of these filopodia-like structures immediately a short time after photoablation. And this makes sense because it's known that the symmetry and the stability of the synapse of a T cell is maintained by a balance of forces in the periphery. And if we inhibit APTO3 complex in a localized way, we can actually transiently destabilize these forces, leading to the symmetry breaking phenotype. So as you will see here, and this is actually very important because the actin cytoskeleton, this underlines how important the actin cytoskeleton is for maintaining the synaptic contact and what can lead to breaking it. And that underlies essentially every adaptive immune interaction. So now the interesting thing that we noticed here is that while we were performing photoablation in the cells, we also inadvertently caused some photobleaching. So we wondered, what would the fluorescence recovery in this area of the cell actually depend upon? And so we figured it could be dark or light actin, or essentially bleached or non-bleached actin molecules, diffusion of APTO3 complex in and out, and the activity actually of the APTO3 complex that we were for the first time able to manipulate in situ using our compound. And I won't go into it in too much detail in interest of time, but what we saw is that different cell types, both non-immune cells as well as our immune cell, which we chose Jurkats for, have different actin recovery profiles upon photoablation in a specific region. And this would indicate to us that the dynamic steady state of actin polymerization and how they sort of assemble their actin machineries and actin assembly in those regions, for instance, when recovering from photoablation in a region, can be different. And we're actively working on some computational simulations that can help us understand this better. And so now I'll conclude by summarizing, saying that we have performed spatiotemporally controlled perturbation of the actin cytoskeleton using our novel compound AZ at the subcellular level, at the multicellular level, whole cell level, and we are also working on cell-cell conjugates and have data now, preliminary data, at the animal level where we have used a C. elegans system. I'd like to acknowledge my advisors, Sudha Kumari, the Kumari Lab members, our collaborators, funding, our facilities, my fellowship, Indian Institute of Science, and of course the organizers of this meeting for giving me this opportunity. Thank you so much.
Hi. I was wondering that in the in cells other than T cells, does your treatment induce cell membrane blebbing? Because you're getting rid of the APTO3 actin.
So I think I have not used it at a high enough concentration to cause membrane blebbing, but in the generic inhibitor, which is CK666, if you use it at a concentration enough to see an effect, you do see membrane blebbing. So I think if I were to sort of titrate the dose of AZ or light treatment, I would see that in cells, yeah.
And the other question is, how long does it take to recover once you get rid of the APTO3? Does it recover in some time? Or?
So in my whole cell perturbation assays, I've seen that up to 20 to 30 minutes, there's no recovery. In fact, which is strange and we don't know why. In fact, the effect lasted for longer and it was a stronger inhibition and after we washed out the inhibitor. But for longer assays, I have looked at the effect for toxicity's sake, and there the effect does reverse, but that was like 24 hours, 48 hours. But in these kind of live assays, I've seen the effect last for up to half an hour without any reversal.
If you could tune the recovery,
because
then that will enable lots of other different studies.
Definitely, and we can also tune the dosage by changing the photostimulation time, the power of photostimulation, and so on.
If you look at motile cells and use this tool, can you sort of guide the motility of the cell by inhibiting? Because this may be particularly useful in the case of neurons.
Yes.
Because you could just decide to.
Definitely. I mean, we haven't tried that, but that's a really good application. In the case of T cells, it is known that they have stop and start mechanisms that depend upon both their antigen search as well, and that dictates how they utilize APTO3 complex at the leading edge. So we could essentially direct cells using this, But I did measure like turn angle and persistence length also when it came to my migration assay, and I noted that there are some distinct changes in that behavior as well. So this could be done in other cell types as well, and hopefully others can try it out. Yeah.
Yes. Question. So do you see a concentration dependence with your compound for perturbing other R3 dependent actin networks within the same cell? Meaning, yes, you showed us that the lamellipodia-like networks are affected, but there are other complexes that are also, other actin complexes that are also dependent on our R3 for their nucleation. So do you know if there is a sort of dose dependence that can allow you to perturb one but not the other? I would imagine the ones you're showing us are the most sensitive.
So T cells are very sensitive to R2-3 perturbation because they use it for more than 60% of their actin at any given time. What I showed about the foci and the lamellipodia, actually there is some data, but again, that's not as well resolved as to how those structures affect signaling differently. So we are looking into how ablating either actin foci, which are important for mechanosensing and force generation, versus the lamellipodia, that's also involved in a lot of maintenance of symmetry, and how those differentially regulate calcium signaling. So maybe a dose-dependent relation could be established there as to if we were to ablate completely versus ablate partially. So that's something that we're looking into.
I was just curious whether you have any toxic effects because of the wavelength of do you observe any toxic effects because of the wavelength of illumination?
So the wavelength of illumination is in the UV range, though I have even tried like 488 nanometers, and essentially it's the power that matters, not really so much the wavelength. But yes, we did see some very interesting effects. It wasn't in the timescale that we used, it wasn't really phototoxic, but we did notice some interesting effects on actin itself upon UV irradiation. You may have noticed that just the control with light, we saw an increase, a burst of actin, and that's something that we're characterizing now as a separate project. But otherwise, I did check for toxic effects in long term. I used a proliferation assay, and the cells were fine within the about 1.5 minutes maximum of the power source that I was using. It was fine, yeah.
Okay, let's thank the speaker.